damn it. Okay. Wow. Hello. It's okay. It's okay. No, I still hear myself. Hello. Hello.
afternoon, everyone. Thanks for coming in, and it's a great pleasure in introducing today's True Talk speaker. So we have this series of True Talk. We we are fortunate that we get to hear to people from all around the world in this series of talks. We started this when we first started this campus back in 2016, and I think this is almost 21st True Talk. I was trying to count how many True Talks have been there. And it's a great pleasure to introduce to you today's speaker, Vanja Sisirak. Vanja is an assistant professor in University of Bordeaux and also a scientist in CNRS immunoconcept in Bordeaux, which is associated with the University of Bordeaux. Vanja did his biology degrees in France, in Lyon, and also did his PhD from University of Lyon, and then did his postdoctoral fellowship in Columbia University, where he worked on SLE. He actually discovered some of the very key concepts in SLE pathogenesis, systemic lupus, which is a systemic autoimmune disease. And now he is extending those observations and those discoveries into other contexts of autoreactive inflammation. Vanja had been a long-time friend, so it's even more great pleasure to introduce him. Apart from being an excellent scientist, he has been a photographer. He had been a DJ in Harlem while he was a postdoc. So you can actually understand he's a very interesting personality and a very student-friendly person. So please feel free to ask questions. As you all know, most of you know that True Talks are supposed to be free-flowing talks for a longer period of time compared to other kinds of talks. The idea is to go to the basis of the science that are being talked about and then asking questions and getting into discussion. So with those said, so Vanja, podium is yours. And this talk is also being hosted by CEFIPRA because CEFIPRA is the Indo-French organization for research support. And because we have one collaborative research with Vanja's lab, as well as Dr. Patrick Blanco in the Department of Rheumatology, University of Bordeaux. So CEFIPRA is also, I can see CEFIPRA has also logged in, and it's being, so they are also recording it. So it will be like broadcast to other national portals through CEFIPRA. And so ask questions. So that is the whole point. Okay, thank you. Thanks, Vanja, for coming over and giving this talk.
To hear me with this, it's okay. So yeah. So good afternoon, everybody, and it's thank you for this very warm welcome in Kolkata. I'm really happy to be here. Like Dipyaman came, this is all possible thanks to the CEFIPRA help that we got this grant together. So Dipyaman came to Bordeaux and visited us and shared some data with us, and I'm really glad that I could come here and meet and see the city of Kolkata and meet the laboratory. And it was really a pleasure discussing with most of the students here. So I will present today my work that was done as part of it during my stay as a postdoctoral fellow back in New York, and so this is mostly on the function of DNases in the regulation of autoimmunity. And later on, I will share with you some of our data that we are exploring about like the role of DNase 1L3 in the regulation of the metaflammation that happens during obesity, which is a project that we are developing in collaboration with the Dipyaman laboratory here. Okay. So let's start. Does it work? Yeah, with the basics. I don't know if, yeah, that works. Okay, so as you all know, probably like one of the important function of the immune system is the detection of pathogens such as bacteria and the viruses. And when the immune system detects these pathogens, it will create initiate an antimicrobial immunity, basically to protecting the host against infections. But so more specifically, this happens, this process is a little bit more complicated. And so usually there are constituents of bacteria and viruses, which are called pathogen-associated molecular patterns, which are recognized by receptors that are expressed by the innate immune system, which are called pathogen recognition receptors. And the recognition will trigger a signaling cascade that is very important in the production of inflammatory cytokines, which are required for the establishment of antimicrobial immunity. As you can see here, one important pathogen-associated molecular pattern is the microbial DNA, either originating from bacteria, viruses, or fungi. No problem. No problem. I move this one. I have mine. So as you can see here, like one of these important pathogen-associated molecular pattern that is important in the activation of the innate immune responses is the microbial DNA, and this microbial DNA can be sensed by certain pathogen recognition receptors that are specialized in DNA sensing. And these pathogen recognition receptors that are specialized in DNA sensing include the endosomal toll-like receptor 9, TLR9, and the cytosolic DNA sensors, which are CGAS and IM2. So the recognition of the microbial DNA will then trigger inflammatory responses and antimicrobial immunity. So while this mechanism is very important because it confers the immune system the ability to recognize a broad range of bacteria and viruses, it comes as a risk for the host. And this risk is because the main source of DNA in our system is our own DNA that's coming from dying cells, and these dying cells can release DNA, and the pathogen recognition receptors that are called pathogen recognition receptors are poor, they poorly discriminate between the microbial DNA and the endogenous DNA. Okay, and so if you have a dying cells that release a huge amount of self-DNA, this can activate these dendritic cells through the same pathway, signaling through TLR9, CGAS, and IM2. And this aberrant recognition of endogenous DNA can lead to the formation or the development of inflammatory syndromes and autoimmunity. So these mechanisms, the system has to prevent this from happening, otherwise we'll be all having autoimmune diseases. And so our organisms have evolved mechanisms to prevent and tightly control the amount of endogenous DNA that we have in our system. And one such mechanism include okay. All right. One such mechanism includes these desoxyribonucleases, also called DNases. And there is a two family of these DNases. There is cell extrinsic DNases that will degrade mostly DNA that is outside in the extracellular space coming from dying cells, and cell intrinsic DNases that prevent an accumulation of DNA within the cells and the aberrant activation of intracellular DNA sensors. Okay. And so these DNases basically prevent this accumulation of DNA, prevent its presence in the cytosol, and this will put a stop basically to any ability to this endogenous DNA to signal through this receptor, innate immune receptor, and this will confer tolerance to self-DNA. Okay? So how we are sure that this works, it's actually there is a lot of genetic studies, and these genetic studies were done on individuals or mice that were deficient for either cell intrinsic DNases or cell extrinsic DNases. Trying to switch slides, it's coming. Oh, all right. So, and these, for example, cell intrinsic DNases, and so two important cell intrinsic DNases are DNase 2, which is mostly expressed by macrophages, and another DNA that is cell intrinsic and present in the cytosol of most of the cells of the organism is DNase 3, and this DNase 3 is also called Trex1. Okay, and so the deficiency of these DNAs is either in mice but also in human will cause an accumulation of intracellular self-DNA. And this intracellular self-DNA is going to be aberrantly recognized by this intracellular DNA sensor, which is called cGAS. This cGAS will reduce or induce a production of type 1 interference, and this will cause fatal inflammatory disorders, which are really severe. And an example of these syndromes is the Aicardi-Gouttiere syndrome or a familial chilblain lupus. Okay, and these are also called these diseases belong to the class of interferonopathies. So pathologies that are induced by aberrant interferon production. So these are really serious diseases that are mostly dependent on this cGAS activation in the cells. And so there are cases also of mutation in human and mice that were found for this. On the other hand, there is these cell extrinsic DNases that I told you that they are degrading extracellular DNA. And there was studies initially done in the early 2000s with the mice showing that DNase1 deficiency is causing an activation of DNA-specific B cells. And these DNA-specific B cells will produce antibodies directed against DNA. These antibodies will create immune complexes with the DNA, and they will deposit in various tissue. And usually the tissue that they deposit is the kidney, where they will cause a kidney inflammation. And so this is basically, these are all the hallmarks of lupus, systemic lupus erythematosus, autoimmune syndrome. Okay, but these studies here that were done with DNase1 deficiency in mice were like, there was one study, two studies actually in Nature Genetics. The first study was in mouse, and so these people did this experiment in how do you call mixed background mouse, and when they purified the background of this mouse, this phenotype basically disappeared, indicating that it was something else that was inducing like this susceptibility. Uh, any okay, it's back. Hello? Yeah, it's okay. So it is back. So it's just to show you that this DNase1, it's not sufficient like to prevent autoimmune diseases, and that there are probably other compensatory mechanisms that like are compensating for the lack of DNase1 to a display of extracellular DNA. And actually there are other family of, this DNase1 belongs to an entire family, and there are other proteins that are associated with this DNase1 that are within this DNase1 family. So there is DNase1 here. So as I say, that this DNase is involved in the digestion of extracellular DNA, but mostly in a naked form DNA. And there is this family is extended through three additional number: DNase1L1, DNase1L2, and DNase1L3. DNase1L1 has a very tissue-specific expression. It is expressed mostly in the cardiac muscle, and and there are some polymorphisms that are associated with a certain cardiac disorders, but like its function in the regulation of extracellular DNA is not very known. It was reported that this enzyme may have maybe not totally secreted, but maybe attached to the membranes of the cells, but its function is relatively poorly studied and unknown. And mostly specifically expressed by the cardiac muscle. This DNase1L2 is expressed mostly within keratinocytes in the skin, and actually it's associated to the DNase1 family as extracellular DNases, but these DNases function at the acidic pH, so it's usually a function when only in endosomal compartment reaching acidity, and it's involved in the degradation of nuclei coming from keratinocytes. While DNase1L3 is an enzyme that is expressed mostly by innate immune cells, and this enzyme can be detected in the circulation, so its distribution is mostly systemic. Okay, and so we were interested in this enzyme DNase1L3 because at the time we started our study, there was a Nature Genetics paper that came out from a family in Saudi Arabia in which there was a loss of function mutation that was detected in those individuals, and this loss of function mutation conferred like induced the development of lupus in this family with almost a Mendelian inheritance. Okay, and so we were thinking maybe this enzyme is very important in the regulation of extracellular DNA, and maybe that was the enzyme that was compensating for the activity of DNase1. And so then what was reassuring is that after this pioneering study that came from Saudi Arabia, there were multiple null mutations in DNase1L3 that were associated with the development of systemic lupus erythematosus. So this is a study in Turkey. This is a study in Italy. There is a study, another was study in Turkey, and there are multiple studies here in France now showing the association of null mutation in DNase1L3 that are associated with lupus or lupus-associated diseases such as hypocomplementic urticarial vasculitis. Okay, and so to add on this, it's a there is not only null mutation which are associated with the DNA with the systemic lupus erythematosus development, but there are also single nucleotide polymorphisms that causes there's one very known, which is the replacement of an arginine into cysteine at the 206 position. And this single nucleotide polymorphism basically reduces the activity of DNase1L3 and prevents its secretion. And it was associated with the development of sporadic lupus in patients that are not like genetically null. And this single nucleotide polymorphism is not only associated with lupus, but other systemic autoimmune diseases such as systemic sclerosis and rheumatoid arthritis. Okay. So it seems that this DNase1L3 is an enzyme that is very important in regulating the extracellular level of DNA and might play a role in preventing systemic autoimmunity. Okay. So just to go a little bit more into the detail, what is the DNase1L3? So it's an enzyme that is also secreted, part of DNase1 family, as I said. And if you measure the activity of DNases in the serum, this DNase1L3 together with DNase1 contribute to most of the DNase activity that is found in the serum. But there is one major difference between DNase1 and DNase1L3. They are highly conserved and highly homologous, but there is one major difference is this C-terminal tail of DNase1L3, and this C-terminal tail is very hydrophobic and it was described to allow DNase1L3 to penetrate membrane. Okay, so the first description of DNase1L3 function was prevention of transfection. So people will put DNA in liposomes and then treat this DNA with DNase1L3, and they will show that this treatment with DNase1L3 was preventing transfection. So indicating that this DNA is able to go through lipidic membranes to digest DNA, which was not the case for DNase1. And also what was shown is that this DNA is contrary to DNase1, is able to digest complex DNA, which is associated in nucleosomal form. So DNase1 will cut where the DNA is naked, and this is what you use usually for example, ATAC-seq experiment. But this DNase1L3 can dispose of very complex DNA when it's associated with the nucleosome without any helper protease activity. Okay. And it was recently shown that this DNA as 1L3s can also digest the DNA that is associated with antibodies directed against DNA, so in immune complexes, so complex forms of DNA. So at that time, we basically wondered, oops, have to come back. So like, what is the function? We generated the DNase1L3 deficient mice, and we wanted to see whether this deficiency can actually cause disease in the mouse model, and can we study what is the function of DNase1L3 and how it might be involved in autoimmune diseases. Okay. So just as an example, there are multiple forms of monogenic lupus in patients. One form of them is the C1q deficiency. But for example, the C1q deficiency, a complement deficiency in individual will induce a development of lupus, but in mouse, it wasn't the case at all. This mechanism was compensated. So it was a kind of a bet, like maybe this, because in animal models, it's really hard to reproduce lupus. It's usually a polygenic disease. So we wonder that. So what we saw initially is in individuals with lupus, there is a development of anti-nuclear antibodies. So these new anti-nuclear antibodies are you usually check them by the fixation of like antibodies which are present in the serum to the nuclei of HEP2 cells. And then you come with a secondary antibody to see whether there's a staining. And you can see when we take the sera of wild type mice and you stain nuclei coming from HEP2 cells, there's no major staining. You just see the nuclei. But when you do that with the knockout serum of the mouse coming from the knockout mice, you see a staining, indicating that there are in the serum of these knockout mice presence of antibody that are directly recognized as the nuclei. So then we wanted to further characterize these antibodies, and we measured by specific ELISA the presence of anti-DNA antibodies. And as you can see, very early at the age, here in gray, you can see these are the wild type animals. They display very low amount of anti-DNA antibodies, but the purple animals here, already at 5 weeks of age, have significantly more anti-DNA antibodies in their circulation. And as you can see, with aging, this level of antibodies against DNA are raising over time. And here is just another experiment. You can measure the amount of B cells that are specific to to DNA. And you can see very early in those animals, either in their bone marrow or in their spleen, you can detect B cells producing antibodies against DNA in knockout mice, which are not present in a wild type animal. So this was just showing that this, similarly to patients, these DNase 103 deficient animals develop anti-DNA autoimmunity. Okay. But and one other thing that usually happens in the lupus development is the deposition of those antibodies in the kidney. And so this you measure by a staining, which is an IgG staining in the kidney. And so here is the anti-IgG staining, which is in red. And as you can see, wild type animals, so these are kidney glomeruli. There are more nuclei in the kidney glomeruli here. And so these kidney glomeruli are clear of antibodies here, but in the knockout animals, there is a clear deposition of antibodies within those kidney glomeruli. And these antibodies actually contribute to the pathology of these kidneys because this is a normal glomeruli up there in the wild type mice. They are tiny, less cellular. And here are like large, these glomeruli become larger, highly cellular in DNase 1L3 knockout mice. And here is the pathological scoring showing that the pathological score of those kidneys in the DNase 103 knockout mice is higher compared to the wild type animals. So this disease is this pathological aspect of the disease and this glomerulonephritis, which is quite mild compared to other lupus model, happens very late in those animals. It happens after 1 year of age. Okay, so in the individuals that are deficient for DNase 1L3, the pathology is quite severe. And some of them may die of it very young, and it affects young kids very early on in the development. In our mice, the pathology is way milder. And so we don't know really why, but we know that humans are not kept in a sterile environment without any infection, while our mice are. And so that's one hypothesis that these mice might, when they are exposed to several triggers or infections, it can accelerate the disease. And this is something that we are studying currently. But we know, for example, if you pump up the level of interferon in these mice with an adenovirus, you will accelerate the disease, and those mice may die further. Okay. Voilà. Okay. So then we wanted, so these mice develop sort of a disease which is resembling loop sort of activity. So we wanted to see what are the mechanisms and what may be dysfunction of this DNase 1L3. It was described to digest liposomal DNA, but what may be the target in vivo of this DNase 1L3? And so we were thinking maybe this DNase 1L3 is affecting DNA that is associated to microparticles, which are derived from cells. And there are microparticles. One of them are exosomes. So these exosomes are very small. They're usually derived from endosomal compartments. So they are actively produced by certain cells. Commonly, they are kind of rich in RNA, but poor in DNA. Okay. And there are also microparticles or microvesicles or apoptotic blade. But this is a complicated field with different denomination of these microparticles. But these microparticles are usually considered as larger. They are up to 1 nanometer, 1 micron in diameters, and they come usually from the outward budding of the plasma membrane, and they're usually originating from dying cells, so they are rich in DNA. So we wondered whether those microparticles can be a kind of a substrate for DNase 1L3, and whether this DNase 1L3 is digesting this DNA. And so to do that, we basically generated microparticles. So we took jerk at T cells, which is tumor cell line of T cells. We induce a cell death of these cells with a drug which is called staurosporin, and this drug is inducing a huge amount of creation of microparticles. We purify those microparticles and expose those microparticles to various recombinant DNases. Okay, so here they're exposed to nothing. So you can see we recover 100% of DNA. There is very little digestion. When you expose these microparticles to DNase 1, you very fully digest this DNA. And when you expose these these microparticles to DNase 1L3, most of the DNA is gone from those microparticles. Okay, and when you get rid of the C-terminal tail of this DNase 1L3, most of the DNA is recovered, so indicating that DNase 1L3 function, in this case, an ability to digest DNA that is associated to microparticles coming from this C-terminal tail. So then we wanted to validate this data in vivo, whether this is the case. So we put, and so to do that, we went to our mouse and we purified. We took the plasma or the serum, and we basically purified microparticles using differential centrifugation. And what we first saw in our pellets is that we wanted to see whether we have intact microparticles and whether those microparticles had DNA. So we stained those microparticles with a DIG, which is a membrane-specific dye, and with vibrant green, which is a membrane-permeable DNA dye. And we could detect that in the blood, in the circulation of these animals, we had intact microparticles containing DNA. So next, we purified the fraction of this plasma containing microparticles and purified the DNA and quantified by PCR using specific primers for repetitive elements of the genome of the mouse. And we could see that the DNA amount per microparticles was higher, way higher in knockout mice compared to wild type animals. And so this was specific to microparticles, because if you look at the total whole plasma level of DNA, the difference is very minor. The major difference happens in this microparticle fraction. And so this is another experiment. We wanted to see how is this DNA, is it exposed maybe also on microparticles, and is it associated? So we isolated microparticles from wild type and knockout mice and stained those microparticles with specific antibodies that are targeted against chromatin. And as you can see here in purple, this is a staining in knockout mice, and here in gray is a staining in wild type mice. And most of the knockout mice present this high level of staining. And when you quantify that, you can see that there are microparticles. There is an enrichment of this staining in the microparticles originating from knockout mice, indicating that basically this DNase 1L3 deficiency is causing a specific accumulation of DNA within microparticles of these animals. Oops, it's a little bit slow. And so we wanted to confirm this data, whether that was true in humans. So we were fortunate enough to collaborate with the Turkish patient, with the individuals persons who did the study in Turkish patients who were kind enough to share with us the plasma of the DNase 1L3 patients. And similarly, from these patients, we could isolate microparticles that were containing DNA and were intact. And then we measured the level of DNA in different fractions of the plasma of these individuals. So here are control individuals, here are DNase 1L3 null individuals. And as you can see in control individuals, so this is the representation of DNA in a microparticle fraction, the soluble fraction, and this is the whole plasma. And as you can see, there is a very poor representation of DNA in the microparticle fraction. It's mostly soluble that is present there, while in individuals that are null for DNase 1L3, the microparticle fraction is the richest fraction of that contains the DNA compared to the soluble fraction. And this is just the quantification of DNA in the microparticle fraction, and it's enriched in the patients that are DNase 1L3 null. And so this is just once again telling us that in humans as well, there's an accumulation of DNA that is specifically happening in microparticles. So then we went back to our mouse, And we wanted to know what is the main source of DNase1L3 and whether the hematopoietic system, like genetics, like studies of microarray at that time or sequencings have shown that DNase1L3 is specifically expressed in a subset of dendritic cells and macrophages. So we wanted to see whether this was true and can we transfer the disease just by transferring knockout bone marrow cells. So we did something which is called bone marrow chimeras. We took DNase1L3 deficient bone marrow and DNase1L3 proficient bone marrow, and we transferred that into lethally irradiated mice. And so basically these wild type mice are reconstituted either with knockout bone marrow cells or wild type bone marrow cells. So these bone marrow cells will give rise to the entire hematopoietic system except the macrophages. And so you basically reconstitute the mouse with a new hematopoietic system. And when you reconstitute wild type mice with wild type bone marrow, they do not develop like the disease. But when you constitute wild type mice with a knockout bone marrow, you can see that the disease occurs. They have autoantibodies, and these antibodies occur. They have the kidney disease, indicating that DNase1L3 is coming mostly from the hematopoietic source. And then our mice is a reporter mice, so we could detect this expression is a specific subset of dendritic cells. I didn't put the data. So we could detect this DNase1L3 expression mostly in conventional dendritic cells and macrophages. And depletion of these cells was also affecting DNase activity in the plasma. So indicating that the source of these DNAs coming from is mostly the hematopoietic system. Okay. So then we were interested in how this DNA that is accumulating extracellularly in microparticle may be activating the innate immune system. And so, as I said initially, there are three main receptors that are involved in the DNA sensing. And so these three receptors are, there's one endosomal receptor. So this endosomal receptor is expressed mostly in innate immune cells, but also in B lymphocytes. And it recognizes DNA coming from the outside world, extracellular space into the endosome and triggers through MyD88 signaling, which is an adapter molecule, inflammatory cytokine production, or in B cell specifically activation. While the other two receptors are intracellular, they are mostly located in the cytosol, and AIM2 when it detects DNA signals through caspase-1 in order to induce the production of pro-inflammatory IL-1 beta and IL-18. This caspase is required for the maturation of these two cytokines. And the other adapter molecule is a STING, C-gas that signals through STING and produces type 1 interferon. So we wanted to know what is the main signaling pathway that is activated in DNase1L3 deficient mice. And so in this case, we were lucky enough to move at NYU at that time, and we got a small package, so I could get a lot of mouse at that time. And so we basically, it's coming. So I just tell you here, so we basically crossed our DNase1L3 deficient mice with caspase-1, MyD88, and STING deficient mice, okay? And so we generated double deficient mice, and what is this showing you here is that when mice are double deficient for DNase1L3 and caspase-1, they still produce autoreactive antibodies against DNA. When mice are double deficient for STING and DNase1L3, they still produce and even more anti-DNA antibodies against anti-DNA antibodies. When mice are deficient for DNase1L3 and knockouts here, they produce antibody. But when you have a double deficiency with MyD88, you prevent actually the production of antibodies directed against DNA. And this was, we checked all the parameters, glomerulonephritis after and other stuff. And this is just showing you that the activation and the production of antibodies in our DNase1L3 deficient mice is mostly dependent on MyD88. So likely TLR signaling. Okay, and so this is we published this paper in Cell in 2016, and this was the cover that we proposed, but it wasn't validated by the reviewers. I think because of the guns. So this is just showing that dying cells are releasing microparticles, which are maybe charged loaded with a chromatin or a DNA associated chromatin, and these microparticles are digested mostly by DNases originating from the dendritic cell and some extent originating from macrophages, and this prevents those microparticles to activate, go be recognized by autoreactive B cells specific to DNA, and thus avoids the production of antibodies. So then we went further, like in a follow-up study, and I will go a little bit faster here because similar experiments several times. But we know that MyD88 was important, but MyD88 signals through TLRs mostly, but it can also signal through other pathways such as IL-1 receptor. So we wanted to make sure that these TLRs are involved in the pathogenesis that we observe. And so this is just a story like with a lot of knockout mice. Oops. So we did a DNS1L3 TLR9 double deficient mice, and to our greatest surprise, in this case, these mice still develop these SLE features. Okay, so recognition, and this receptor is known to recognize DNA mostly. And so we had mice that were like perfectly fine. There was no big major change in them, and so they still develop autoreactivity to DNA and features of lupus in these mice, indicating that tRNA deficiency does not affect SLE development. So then we were okay, maybe something is happening because of TLR7, because TLR7 is a receptor that is important in the recognition of RNA, but there are also recent studies that are showing that TLR7 can recognize deoxyguanosines, which are kind of degradation products of DNA. So we wonder maybe like this can be through this phenomenon, but nothing happened. Like TLR7 deficiency did not affect SLE development. So then maybe we said it's compensated. They are each compensating each other. So what we did is a triple knockout at that moment. So we basically, oops, it went faster in this case. So the triple knockout, and in this case, boom, DNA is like there was no lupus. So we were like, so this is a surprising study that we are still trying to understand. We don't know how it works. But it's interesting that the double deficiency of those two receptors was totally sufficient to prevent all the syndromes of autoimmunity in those mice. And since these results were kind of weird for us, we wanted to confirm that on our side. So we did another deficiency of DNS1L3 in an ONC93-1B mice. So ONC93B1 is a chaperone molecule that is required for the transport of TLR7 and 9 to the endosome, but also TLR3. So it's basically when you have a knockout of this protein, you could totally abrogate all toll-like receptors that are located in the endosomes, and we also prevented the disease pathology in this case, so which made us a little bit more confident about our results. So then when we went downstream and see what's happening downstream, whether these TLRs are mostly involved in like type 1 interferon production, and interferon is a cytokine that was previously described to play an important role in the SLE pathogenesis. Most of the SLE patients present an interferon signature, and recently there are even anti-IFNAR receptor that are used as a therapeutic therapy in those patients. So we wanted to see that, and in our case, when we crossed DNS1L3 with mice which are deficient in interferon signaling, we had a mixed phenotype. Those mice were like there's an initial break of tolerance towards self-DNA. So our mice are still producing antibodies against DNA, but these antibodies do not decrease as much as in a DNS knockout mice. So there is a less exacerbation of the pathology, and there is less kidney disease and other features of the disease, indicating that this IFNAR is not required for the break of tolerance to DNA, but is exacerbating the disease. Okay. And so then we want to see more like cellular constituents that are very important in the pathogenesis of SLE development in our DNS1L3 deficient mice. And so it was shown that the plasmacytoid dendritic cells are important producers of type 1 interferon. And actually, together with Dipiaman, we did a paper when we deplete these plasmacytoid dendritic cells in various mouse models of lupus. We were able to prevent the disease, and and other papers have shown that this interferon coming from plasmacytoid dendritic cells is important in contributing to lupus pathogenesis in different models of lupus. So we wondered whether these PDCs are important in our model, and so to do so, we were in Boris Race's lab. So he had mice that are TCF4 deficient. So this TCF4 is a transcription factor. It encodes the protein called E22. This E22 protein is required for the development of PDCs. And when you have a haplodeficiency of this transcription factor, you reduce the amount of PDCs that are present in the bone marrow and in the spleen. And when you stimulate those mice with the TLR9 specific ligands, because TLR9 is specifically it's expressed highly in PDCs, you prevent you totally abrogate the capacity of these haplodeficient mice to produce interferon. So these basically indicate that these TCF4 haplodeficiencies are altering PDC numbers and totally impairing their function. So we crossed our DNS1L3 deficient mice with these PDC deficient mice, and we basically got the phenocopy phenotype, like the same phenotype as the IFNAR mice, which is these mice produce autoantibodies, but their production of autoantibodies is not exacerbated over time, and they don't have a full disease, indicating like that mostly not suggesting basically that PDC may be the main source of type 1 interferon that is contributing to the disease. The reviewers ask us that, like, you are not directly showing it. But so we did some experiment where you can target PDC production of type 1 interferon using an antibodies, using Siglec-H. We did that, but it's very difficult because it's a very costly experiment. Our mice model takes a year. So if you have to treat your mice model for a year with an antibody, that costs a lot. So we did a certain amount of time, and we were showing that with this certain amount of time, you could reduce an antibody production, but it wasn't like super important, but like we couldn't prove it formally. And so finally, we were interested in the implication of other actors that are involved in the lupus pathogenesis. So as I told you before, there is these PDCs are producing interferon. These interferon are important for the differentiation of B cells into plasma cells producing antibodies. But another factor is very important for the B cell differentiation in antibody producing cells are T cells. Actually, T cells are providing help to B cells, and mostly through the interaction of CD40 ligand with CD40 or B cells to favor the differentiation into antibody producing cells. But there are also B cells that can be turned on and basically produce antibodies in absence of the help of the T cells. So we wonder if in our case the T cell response is required for the development of lupus. And so when we knocked out CD40 ligand and CD40 interaction in our DNS1L3 deficient mice, we prevented the disease, which was indicating that T cell help is absolutely required for the generation of autoantibodies in our mice. So now we're going to go a little bit further into the biology of B cell responses. So usually T cell dependent B cell responses are occurring in two main spots in the lymphoid organs. So there is the classical B cell response that occurs in the B cell follicle. It's called the germinal center reaction. In this case, a specific subset of T cells called T follicular helper cells, through their production of IL-21 and interaction with B cells, will induce B cell activation and differentiation into long-lived plasma cells. And in this germinal center, there's something else that's happening. It's a class switching. So your B cells will be switching classes and express different kind of antibodies or isotypes of antibodies, but they will be also a somatic hypermutation, which means that these antibodies will be mutated highly to recognize very well their target. So okay, so they will be highly specific. And so the response is usually like germinal center lead to plasma cells, which produce antibodies which are specific and highly mutated. The extrafollicular T cell response, B cell response can also happen, and this extrafollicular B cell response occurs outside of the follicle and leads to the activation of short-lived plasmablasts, which are producing diffuse affinity and less hypermutation, okay? So we wanted to see which one of the response is happening in our animals. And so what we were fortunate is when as I told you before, when we crossed the TLR7 mice with our DNS1L3 knockout mice, there is still autoantibody production. So our mice are still developing autoreactivity. But one thing that was already described in the literature is that TLR7 deficiency totally abrogate germinal center B cells. And so we verified that by flow cytometry. We don't have the germinal center B cells. We verified that by immunostaining. Here, GL7 is a specific kind of staining of germinal center within the spleen. Wild type mice have it. The DNS1L3 knockout mice have it, but wild deficient mice don't have it. So that was a clear indication in a way for us that even in absence of germinal center, we have autoreactivity, indicating that this our T cell dependent B cell response is mostly happening outside of the germinal center. And we also sequenced the B cells from our mice, and we saw very little somatic hypermutation in those, indicating that this was not the case. And this is just a staining showing you, so GL7 is a marker of germinal center, CD169 is a marker of marginal zone macrophages that are usually surrounding B cell follicles, and CD138 is a marker of plasma cells or plasmablasts, B cells that are differentiating. And as you can see, knockout wild type mice don't have so much plasmablasts, little germinal center, but when you look at here, knockout mice have a lot of plasmablasts. Most of these plasma cells or plasmablasts are located outside of the center, indicating that this is mostly something that is happening out of the germinal center. So what time I look? Okay. So I'll maybe I'll go just fast on that. This is just showing we wanted to connect our story and what is the function of type 1 interferon and PDCs in this context. And this slide is just showing in absence of interferon, you have less plasma cells in the outside of the germinal center. And this was quantified here. And this is in absence of PDCs, you have the similar response. And we did some in vitro studies indicating that both these PDCs and the interferon that is derived from this PDC is important for the differentiation of naive B cells or into plasma-producing cells, indicating that these PDCs and interferon derived from them are quite important in the maintenance of these cells. So this work was mostly done in Boris's lab, where I was there, and together with postdoc Chetna Soni, that we were working together. Actually, Chetna is coming back to India soon, she came back. In which city again? Lucknow, and started her own independent career there. And so this is just a further this is a study that we published recently, showing like that we further refined our understanding of the DNase1L3 function and how the pathogenesis occur in those animals. And so here is just showing you that the microparticles, in absence of DNase1L3, will activate toll-like receptor signaling in PDCs, but also in B cells. And this toll-like signaling in PDCs inducing interferon production, this interferon is important also for the activation of B cells. And this activation of B cells require total T cell help to lead to the production of antibodies. And so like blocking actually these receptors is something that can be a valuable tool for the patients that are deficient for DNase1L3. And actually, I was talking with people like in Bordeaux who discovered a small family, like two sisters coming from Syria that had this mutation, and their disease were always exacerbating. But when they gave them plaquenil, so it's hydroxychloroquine, which is something that blocks like the acidity of the endosomal compartment, so the function of TLRs in the endosomes, these patients do better. Okay. And so we have some other further studies that we are working on. That so we did experiments just to what we are heading now with Boris, and we did experiments at that time, and we are finalizing this manuscript here. We generated DNase1L3 deficient mice that are also RAG1 deficient. So these mice don't have T cells and B cells. And in those mice, we can transfer either wild type T cells and B cells or knockout T cells. They genotype, basically when you transfer T cells and B cells either of wild type or knockout nature, they will be, when the background of these mice is DNase1L3 knockout, they will be producing autoantibodies. But the important aspect of it is then you can play with this system. And so we did use B cells that are deficient for TLR, and we found also that the TLR specific deficiency in B cells is absolutely important in this phenotype. Okay, so it's PDCs are important, but this signaling that is happening in B cells is critical. Okay, so I'm going to switch gears now a little bit, and I'm going to be talking to you about our project that we developed here with Dipyoman in Kolkata. So this project was supported by the CEFIPRA, which is a French a Franco-Indian association supporting international collaboration, and this work was mostly to understand what may be the function of DNS1L3 in the regulation of extracellular DNA in the context of obesity and inflammation that is induced in context of obesity. So on the French side, this work was mostly done by Amandine Ferrier, who was a PhD student who just graduated recently and went back to the hospital. She was an MD PhD student, and this work is being extended now by Anaïs Rouberti, who is another PhD student working in the lab. And so we do that in collaboration with Dipiaman, who is mostly providing intellectual contribution and also a lot of like patient study here. And in the lab here is Purvita, who is working on that aspect. And so why we got interested in this metaflammation? So I'm going to just give you some little bit rationale and why we came to that study. So the first thing that I want to emphasize here is that during obesity, there's several things that happen. So you have an exacerbation of the adipose tissue, and this exacerbation of the adipose tissue will also lead to a reorganization of this tissue and mostly a total change in the immune infiltrate. So usually in the lean adipose tissue, you have the presence of M2-like macrophages, which are macrophages which have anti-inflammatory function, and when obesity happens, there is a hypertrophy of the adipose tissue, but there is also a phenotypic switch of macrophages that occurs. Is that M2 macrophages are replaced by M1-like macrophages, which are macrophages mostly producing inflammatory cytokines such as TNF alpha or interleukin-1 beta and other factors. And these macrophages here contribute to the inflammation of the adipose tissue. And this inflammation, the production of these cytokines, will lead to something called like a metabolic syndrome, which will impair like insulin signaling, induce insulin resistant, glucose intolerance. And this will contribute to a lot of complications that are associated with obesity, such as type 2 diabetes or cardiovascular issues. Okay. And so there's this switch of macrophages that occurs. And so it, like we say switch, but it's mostly a recruitment of macrophages that are coming from monocytes that will infiltrate the tissue, and they will right away acquire also M1-like phenotype. So there is an inflammation, and this inflammation during obesity is responsible for the complications that are associated with obesity. So recently, there was a lot of studies, including a study here from Dipiaman laboratory, that is showing that during the obesity there will be like a lot of cell death. This cell death occurs because there's an exacerbation of adipose tissue. The adipose tissue at some point the adipocytes from the adipose tissue will die. Hepatocytes are because there is liver steatosis, so this inducing hepatocyte cell death. And there is also a process of netosis. We don't know what are the triggering factor, but that are inducing a cell death of neutrophils. And this cell death of neutrophils, all of these forms of cell death will lead to the release of endogenous DNA. So this DNA can be oxidized. This can be bound to microparticles. There is also mitochondrial DNA that is released, or genomic DNA. And so it was described that obese individuals have an elevated level of circulatory DNA, and that these elevated levels of circulatory DNA in obese individuals was highly correlated with the complication associated with the pathology. So indicating that it must be a factor that is contributing to the disease. And so then there were follow-up studies on this, indicating that this DNA that is accumulating during obesity can be recognized, as I said before, by endogenous like pathogen recognition receptor, such as CGAS and TLR9. These engages, as I said previously, like different signaling pathway. This signaling pathway lead to the activation of transcription factor that are required for the production of inflammatory cytokines. And these inflammatory cytokines lead to metaflammation and contribute to the complication of obesity. So it was shown that deficiency of toll-like receptor 9 during obesity will reduce metaflammation and ameliorate the metabolic parameters of the animals on their high-fat diet. And there was here a pioneering study by Dipiaman actually, who showed that the DNA originating from adipocytes, which is associated with a protein called HMGB1, I won't go into the detail of that, but this HMGB1 and TLR and DNA that is released by adipocytes can be recognized by toll-like receptor 9 in plasmacytoid dendritic cells present within the metabolic tissue, and these plasmacytoid dendritic cells will produce interferon, and this interferon, Dipiaman has shown in his laboratory, is very important player in the switch of macrophages towards a more pro-inflammatory phenotype, and so thus contributing to the obesity-associated complication. So given these DPMN results and others people results indicating that there is an accumulation of extracellular DNA and that this extracellular DNA is important in the activation of innate immune responses during obesity, we're wondering like what is the function of DNase 1L3? It should be there in a way to prevent these kind of events and control the levels of extracellular DNA. And there was some interesting observation that DPMN made here and contacted me. So we developed this project together is that when he put his macrophages under condition that they will become more pro-inflammatory, their expression of DNase 1L3 will be reduced. Okay, so we wondered in this case what may be the function of this DNase 1L3 in regulating the ability of endogenous DNA to stimulate immune responding during obesity. So we went to our mouse study. So we took our wild type mice and knockout mice, put them on a normal diet, which is regular diet of the mice, or high fat diet. So we use two types of diet, but this is the result on a 45% of fat diet. So this is mostly a diet that is associated like with a kind of related to the Western diet. Okay. And so we put those mice on diet for 12 weeks. We measure the body weight weekly. At 6 weeks and 12 weeks, we do a glucose tolerance test, and we take blood for measuring the circulatory glucose level, insulin level, and other parameters such as triglycerides, like and liver enzymes, so aminotransferase. And at some point we sacrifice those mice and we check their organs. And so this was just our first like initial observation, and we repeated that over like with different diets using different animals, sex of animals. So these are all male mice because male mice are usually more susceptible to obesity and obesity associated metabolic syndrome. And as you can see, our normal mice, like wild type and knockout mice on normal diet here, they are not different between each other. But when you put mice on high fat diet, you see they gain weight, and you see that our DNase 1L3 deficient mice gain way more weight than the knockout mice, the wild type mice here in the light blue. And so when you measure the body weight gain, so which is the amount of grams that these mice took from the beginning of the experiment, you can see that this is significantly higher in our knockout mice compared to the wild type mice at 6 weeks and 12 weeks after the start of the high fat diet, indicating that our main mice are getting DNase 1L3 deficiency exacerbate like the weight gain. And so then we wanted to see where this weight goes or comes from. So we took the adipose tissue and we showed that the mass of the epididymal or visceral adipose tissue was higher in our DNase 1L3 deficient mice compared to the wild type mice. And when we did histological analysis of these mice, you can see the adipocyte size is increasing, open high fat diet, and this size is highly increased in our knockout mice. And this is, for example, the measurement of the adipocyte perimeter, which is significantly higher in our wild type knockout mice compared to the wild type mice. So DNase 1L3 deficiency actually exacerbates the adiposity. And we saw that also by using a magnetic resonance study. And we show that the adipose mass is way higher in our knockout mice compared to while the lean mass is remain the same. So then we wanted to understand the mechanism why this happens. Like, what is there something weird in the behavior of our animals, and do they eat more or don't, etc. So we put those mice in individual metabolic cages and follow them for some time. So this is just mice on regular diet. As you can see, this is their activity. The activity in black of knockout mice compared to wild type mice is quite similar. And when you look at their energy expenditure, it's quite similar. Here is the respiratory exchange ratio. So it's the amount of oxygen taken to the amount of CO2 that is produced. And you can see that there is maybe some changes during the daytime, which is indicating that these mice may have some more activity at daytime than the knockout mice, but nothing very severe. And so then we looked at the food intake. So this is the food intake or during the day, and you can see that mouse they are active during the night and eat during the night, but there is no major differences between knockout and wild type mice. And during the day, their food consumption is kind of similar. There is maybe more peaks for DNase 1L3 knockout mice, but we'll go that to the later. And this is the cumulative food intake, and there is no major differences between the two mice. So there are not like major differences in the amount of food that they ingest. The only difference that we observed is that our mice at daytime will have more meals. And so there is a number of meals is higher and the interval between the meals is lower. So it's indicating that our mice are kind of snacking. But the total amount of meal is at the end similar. So there's no major differences. So we don't really, maybe there's something else happening, maybe something at the level of intestinal permeability that is messed up. So this is something that we are actively studying now. So then we wanted to see whether, like after like checking like the weight of our mice, is like, are those mice susceptible to more metabolic syndrome, and do they have metabolic changes that occurs? And one way to measure that is a glucose tolerance test. So you basically inject in your mice glucose, and you see how this glucose is cleared by the system. And what you can observe is here that the glucose intolerance is increased upon a high-fat diet, basically, and it's significantly higher in mice that are knockout for DNase1L3. And so this is at 12 weeks, and we did that. Like the results are more striking when you use a diet which is more fat. And this is the area under the curve. It's not significant, but significant, but it shows like a trend toward a more glucose intolerance in our DNase1L3 knockout mice compared to the wild type mice. So then we also look at the level of fasting glucose. This fasting glucose is increased in our knockout mice compared to the wild type mice, and the insulin levels are not significantly increased, but there is a trend. And when you measure the score of insulin resistance, which is this HOMA ratio, which is the amount of insulin multiplied by the amount of glucose in those mice, and you divide that by 4 or 5, you see that our mice are like more insulin resistant knockout mice compared to the wild type animals than they, and indicating that these mice are more susceptible to metabolic syndrome. Up, and I talk too much, I'm sorry. And this is just, I'll go okay. So we also check the liver of these mice, and so what we first show is that in their circulation there is an increased amount of ALAT, so which is alanine aminotransferase, which indicate that the liver is malfunctioning. And so then we went to the liver of these mice, and we saw that there is actually that at high-fat diet, which is mild diet, there is very little steatosis that is developed in the wild-type mice, but these steatosis occur in the knockout mice, and histologists analyze the score of steatosis that is happening in our mice, and the steatosis is significantly increased, indicating that DNase1L3 deficiency also increased the susceptibility of our mice to develop a liver steatosis in response to high-fat diet. So these are all complications associated to obesity. So then we wanted to see, well, why, what is happening? So we went and took the adipose tissue, the visceral adipose tissue from our animals, and we checked the infiltration of different immune cells to see whether there are some changes in the immune infiltration. And as you can see, there is an increase always of CD45 cells, which are all immune cells in the adipose tissue. And this increase is like in the number of cells was way higher in our knockout mice compared to the wild type mice. And this infiltration of immune cells mostly correspond to macrophages, which are here CD40, CD64 positive, CD11B positive. They accumulate in the adipose tissue of these mice. And when we check the phenotype of these macrophages, so there are two types of macrophages that I told you that are present in the adipose tissue. There are the M2-like macrophages, usually present mostly in abundance in the lean tissue. They express CD310B. And there is these macrophages that are expressing CD11C that are mostly M1-like macrophages and pro-inflammatory. So this is a wild-type high-fat diet. And you see that there's very little M1 macrophages, but still more than in the normal diet. But when you use the our knockout mice are accumulating more inflammatory macrophages. So there there's likely more inflammation in the adipose tissue of those animals. And this was significant at the end of the day. Okay. So maybe I'll go fast. So this was just to show you. So this is the data of mice. Somehow our mice are more susceptible to metabolic syndrome and complications that are induced by obesity. So DNase1L3 deficiency or DNase1L3 is likely regulating the immunostimulatory potential of endogenous DNA and preventing it to aberrantly activate inflammatory responses. So these are mouse studies. So we wanted to see whether that was there was some relevance to our studies into humans. So we collaborated here with Diploman, but we also collaborated with the Department of Nutrition at the University of Bordeaux, and we got access to the blood of at least 40 patients that were obese, and we isolated the different the DNA of their plasma. And so we isolated DNA from different fractions. And so here's the whole plasma fraction. And you can see here also that the DNA amounts are increased in the whole plasma fraction in the obese individuals compared to control individual. When we look at the microparticle fraction, we didn't see major differences here, which is something interesting because like maybe like we'll see later why it matters, but this is usually a privileged target of DNS1L3. And when you look in the soluble fraction, we saw that an increase was high. But so then we were surprised by this data that there was no differences in here in the microparticle fraction. So we went further and we show in this case that the number of microparticles that we detect in obese individual was kind of significantly reduced. So if you then normalize the quantity of DNA contained in the microparticles per number of microparticles, you see that our microparticles have more DNA than in the obese individual compared to healthy donors. Okay. And so then we checked different parameters of these, and we correlated the levels of the DNA with different pathological parameters of these patients. And there is a weak correlation, but significant correlation with the body mass index. The more weight you gain, the more DNA in the circulation you have. And there is a better correlation with insulin levels of these patients. So the more DNA you have in your circulation, the more insulin like you have, and the more like you are basically insulin resistant. So indicating that this amount of DNA is associated with pathological features, which was something that was previously reported as well. And so then we also were fortunate enough to get access to another collaboration with the Nutri-Neuro department. So we have a neuro department working on nutrition in our institute, and they were able to provide us plasma of the patients who were obese and who underwent bariatric surgery. And we got this plasma of these individuals 6 months after bariatric surgery, where these patients drastically lost weight. And what we saw is that prior surgery, they have high amounts of DNA level in the whole plasma fraction, and this is significantly reduced after surgery. And this was the case, like mostly soluble DNA. Like in this, we couldn't count the amount of microparticles because we didn't have enough of the plasma of these individuals. But in the fraction, we didn't see in this microparticle fraction, we didn't see major differences, indicating that when these patients lose weight, their quantity of DNA is against reduced in the circulation, indicating that these DNA levels in the circulation are actually strongly associated with the pathological severity. And so then we wanted to see what's happening, like why this DNA is increased, the DNA circulatory DNA is increased in the patient. Is there some alteration in the overall expression of DNS1L3? So for that, DNS1L3 it is a tough enzyme to work with because there are no specific antibodies nowadays. Nobody has done like a good job making some. Now, I'm sure they're going to come soon because some lupus patients were described with producing anti-DNS1L3 antibodies. But so far, there's no good antibodies. So what we did is a technique called flow fish. Is like a fish technique. You use a probe that is directly detecting your RNA of DNS1L3. So it's checking the level expression of mRNA. But this probe is a fluorescent probe whose signal you can amplify and then check its expression by flow cytometry at the same time as your marker. So you can check at the individual cell level the expression of DNS1L3. And so we did that, and what we observed is that in the circulation, these are different types of cells, T cells, B cells, NK cells, etc., that are present in the circulation. And what we see is that DNS1L3 has a very specific expression to PDC. Most of the other cells do not express it in our hands. But when we compare between obese individuals and healthy donors, we did a lot of patients in this case, but we were surprised to see that there is no modulation in the expression of DNS1L3, at least at the level of the blood. We don't know what's happening in the tissue, and there is some data from here in the laboratory that DNS1L3 may be regulated specifically in the adipose tissue and might have a different expression profile. And so if the expression is not messed up, what's happening with the activity of DNase1L3? So for that, we isolated the plasma of these obese individual and normal individual, and did what is called a SRED assay. It's a single radial enzyme diffusion assay. It's basically you make a well in a gel that is already loaded with DNA, and this well you fill it up with plasma, and or serum mostly, and as the serum like progresses, it will digest your DNA. It will create a halo. And so you can measure the halo. This is the activity where the DNase one or the DNAs have digested your DNA. And so this is just a positive control. When you put DNase one, you have digestion. When you don't have enzyme, you have no digestion. And so here is obese individuals, and here's healthy donors. So you can see the halos are a little bit like smaller in obese individuals, indicating the reduced activity, but this was very hard to quantitate. So then we switched method, and we use a picogreen assay. Is basically where we take an amount of DNA, and that we put in presence of plasma of or this is mostly plasma in this case of the patients or healthy donors, and then we come up with a fluorescent dye which will bind to double-stranded DNA, and which will basically be the proportional to the amount of DNA that is remaining after the digestion, and then we calculate DNA's activity in the plasma of these individuals, and as you can see, DNA's activity is highly reduced. It's reduced in obese individual compared to a normal individual. So these are some data that are indicating that overall expression is not affected, but mostly the maybe the enzyme activity is somehow regulated by some modification that occurs in those individuals. And so this is just to finalize that here is the DNA's activity is highly, it's very well correlated with the levels of the DNA that is present in the plasma. The more activity you have, the less DNA you have in the circulation. And this DNase 1L3 activity is like inversely correlated with the body mass index. The more weight you have, the less DNase activity you have. Okay, so these are something pathological parameters. And so this is just to finalize this story. So this our story is saying that during obesity, there is an accumulation of extracellular DNA, that this extracellular DNA is likely activating the innate immune cell production. We have to do more work in these aspects to see inflammatory activation, and that DNase 1L3 may be an important aspect in controlling this amount of extracellular DNA during obesity. And so what we are now doing like is checking what are the main sources of DNase 1L3, whether this DNase 1L3 is coming directly from dendritic cells or macrophages, because macrophages are a big player in obesity phenomenon. And we want to know what are the mechanism of action of DNase1L3 in metaflammation. So we are checking the dendritic cells and macrophages from the adipose tissue or the liver, whether they are more they can produce more inflammatory cytokines in absence of DNase1L3. And one thing that we are currently doing, we actually transferring DNase1L3 into animals, inducing its overexpression using adenoviral vector as a therapeutic like potential. Can we reduce basically the inflammation that is induced by obesity by adding DNase1L3 in those animals? And so this is something that we are currently doing and trying like to push prior the publication of this study. So I went over time, and I'm sorry. And I thank you for your attention. And so this is just an acknowledgement. So most of the initial work on lupus was done together with Boris, with the great collaboration with a lot of students that I work with. These are graduate students that I work with, Benjamin Sali, Ali Rashid Faruqi, Lee Serpas, Chetna, that I worked a lot with her at that time. And so these were our collaborators that helped us either get the patients or like tools which were essential for our studies. And so here's most of the work that was done about obesity was done in Bordeaux, mostly by Amandine Ferrière and Anaïs Rouberti, and with the help of other members of the lab. And so this was done in close collaboration with Dipiaman here and Prabita. And actually, Prabita came to Bordeaux as a Raman Sharpak fellow, supported by this program. And this program is actually supported by the CEFIPRA, where we got additional funding from the French National Agency of Research and actually the University of Bordeaux. And we are continuing this work on this. So thank you very much for your attention, guys, and thanks a lot for. I'll be happy to take any questions that you may have if you're not too tired of it.
So in your first part,
Uh-huh.
so DNA is I1L3.
Yeah.
So is it actually working on the microvesicle containing DNA, or like most of the cases when there is a microvesicle and the DNA is inside,
Mhm.
right? So
Mhm.
first question is that how it is working on the DNA,
So
whether when it is.
we don't know actually the mechanism of action. We know that this mechanism of action requires its C-terminal tail. We don't know. I think we know that when we digest, these microparticles, we can prevent this staining with the antibodies. So we get rid of the DNA that is associated to the membranes, or like extra, like that is outside, exposed on those microparticles. But we also measure the DNA within inside,
Yeah,
and there seem also to be a
yeah,
reduction.
yeah.
But we don't know how really
So
like.
is there any I mean, I'm just wondering, is
Yes.
there any specificity, or is there anything that it only targets microvesicle containing DNA rather than that naked DNA?
Yeah, I don't know. Or any so this so the it can digest naked DNA as well. This enzyme can do it, but DNase1 is better than that. It can do it, but this enzyme has like also. So it seems that something is related to the C-terminal domain, and why is that? We really don't know. So some people had said that this C-terminal domain looked at like a lipid penetrating domain, and that it might play a function, but then yeah, there should be also a mechanism that would inactivate this enzyme if it enters the cells, like at some point, like it is. So we don't know really the biochemical aspects of these enzymes are only being now a little bit known, and there is a paper, a structural paper that just came out like this year, trying to explore that, but nothing yet is sure about that.
Okay. And another question is that, as you have shown that this T cell help is very much required.
Yeah.
So I hope it's a CD4 T cells, right? Mostly.
Yes.
But have you tried
CD8
CD4
depletion?
versus CD8 depletion? Yeah.
No, we didn't try, but like we believe it's it's mostly.
And do you have any idea of what kind of T cell it is? I mean, have you done any single cell sequencing of this to identify the T cell signature?
So there is we haven't done any characterization of T cells. We have some studies that are showing that some T cells accumulate in those DNase 1L3 deficient mice, and those T cells express very low level of CD62L, and they express very low level of PSGL1, and so these T cells may correspond to some extrafollicular T cells, and they express higher level of ICOS, CXCR4. There is some aspects like this, but we don't know. We haven't depleted these cells. Well, like causative event of these cells, we don't know. We don't believe it's T follicular helper cells because they don't seem to be bothered. Like they don't accumulate, and and the germinal center reaction may not be the important in this mouse model. But further characterization of these T cells, we don't know. And these T cells maybe they were described like these kind of extrafollicular T cells are in lupus patients in the circulation as what a T peripheral helper or TPH cells, which share similarities, but we are not sure. We didn't do a full characterization of that.
So one last question before we take the question from online. Yeah, there is some question there. So last question is that, so what triggers the down regulation of DNase 1L3? Because in the inflammatory settings, you have shown that there is a down regulation of, or Dipo and Ganguly's lab have shown there is a down.
We didn't see like actually we don't know. So it seems that it's more expressed in obese tissue. So
it's more or
it's there in our case, like in the peripheral blood, like there's no differences, but in the adipose tissue is maybe more expressed. And then there are some description, but like it was less expressed in the in vitro settings, and it was in human. In this case, in our case, we know that certain inflammatory signals will induce this DNase 1L3, but then maybe there is some other aspects that that happens within the circulation that can affect the activity directly of this enzyme, and this can be the acidity. This can be other aspects. There's maybe too much lipids or other aspects of stuff. So we are now doing some kind of experiments which are called or they can be autoantibody. So in lupus, there were autoantibodies that were shown to prevent the activity of this enzyme. But so now we are trying, because some obese individual can develop autoreactivity. So we're trying to see whether there is antibodies for this enzyme.
Okay.
But also we are trying some experiments where we take obese plasma or serum and we add DNase 1L3 to see whether there's an inhibition of the activity and stuff like that, and then maybe see what factors may be involved in that.
Okay.
Yeah, for sure.
So, next question. So, is there any affinity for DNase 1L3 towards genomic DNA or mitochondrial DNA?
So these are like excellent question. Like we don't know. We don't know much. Like it's very like this specificity is very difficult to put because we don't have antibodies directed to this enzyme and stuff like that, and to study how it works. We know that it digests genomic DNA. Mitochondrial DNA changes we haven't seen so much in the circulation, so it seems mostly targeted towards genomic DNA. So then there are some. So we collaborated with a person in Hong Kong who is specialized in circulatory DNA. It's a person called Dennis Loh, who is very important. Like who did like this quantification of maternal DNA for the determination. Like you can determine the sex of the baby according to that. And so he did some sequencing of the DNA of our animals and also of the individuals that were deficient for DNase 1L3. And so what he has observed that there, like usually the most dominant form of DNA that is present in your circulation is a mononucleosomal form. So 160, 166 base pair or something like that. And so this form of DNA is fully affected by DNase 1L3 deficiency. But what is increased is the polynucleosome, like a kind of larger fragments of DNA. So what he claims, together with us actually, is that there is less fragmentation of nucleosomal DNA that's coming from apoptotic cells. And there is another aspect of that, it's his sequences. So he has the sequences and he sees some end motif enrichment. So in absence of DNase 1L3, you lose the end motifs like CC, T, CCA. So it might have some sequence specificity and stuff like that. But then there is an interesting question, like maybe this enzyme is also involved in pathogenic, like in pathogenic conditions and stuff like that.
Lupus.
Like, or like also infectious condition. What happens with infectious DNA? And so here also we have like diverging results. If you stimulate your cells with certain CPGs or viruses, your DNase 1L3 deficient disease will be producing certain times like less interferon or certain other things like certain inflammatory cytokines. So it depends maybe on the quality of the DNA matters a lot in this case. And actually we have a very interesting observation that is like messing us up. It's in the tumor environment. So it was shown that, for example, when you do chemotherapies or radiotherapies, this will release a huge amount of DNA, and this DNA is required for the activation of immune responses against the tumors. So we're thinking, what happens like if we delete this DNase 1L3, maybe we're going to increase this rate and boost anti-tumor immunity. And we had the totally reverse phenotype. So indicating that maybe types of cell death that you are inducing or origins of this DNA may matter. So
this leads to the second naive question. So have you ever checked whether DNase 1L3 has any interfering effect on DNA release itself?
So yeah, some people have done that. Like there's a Japanese lab that is working and saying that this DNase 1L3 may replace for this caspase dependent DNA that is involved in the DNA fragmentation during the cell death process. So, but it's hard to conceive this information because it has a very specific expression profile. Why would an enzyme be expressed specifically in macrophages and dendritic cells to be involved in the fragmentation of DNA after apoptosis? So it's kind of weird, but there is a recent study that has shown something interesting. So there is this species of DNA, but that we don't know so much about it, is this circular extra chromosomal DNA that can accumulate in the cytosols. And this circular extracellular chromosomal circular extra chromosomal DNA that accumulates was suggested to be maybe DNase 1L3 dependent. One paper in Nature, something interesting for us because this DNA, circular DNA that is coming out of the nuclei is genomic in this case, but was involved in the activation of immune responses in the tumors. Something interesting to us.
Thank you.
Can we take the online question? Yeah. So Debu and Dr. Arindam Talukdar and Dr. Debu Pradubishas. So Dr. Debu, yeah.
Can I ask you a question? Yes. Can you hear me?
Yes.
Okay.
Yes, we can hear you.
Yeah, so it seems that the DNA's 1L3 knockout would make a bigger fragment of DNA, right? That's what you say.
It's important in the fragmentation of DNA that is extracellular. Yes, there is less fragmentation of circulatory DNA in DNase 1L3 deficient mice and humans.
So, what are the chances that this bigger, fragment of DNA getting into some kind of cellular environment within cells and kind of acting as a sponging mechanism for the, let's say, for me interested in transcription factor binding to that DNA and then ultimately, reducing the overall pool of the transcription factors which are required for your responses that you are looking into?
I haven't thought of that. It's an interesting question. I don't know. What is known, at least, like I don't know anything like about regulation of transcription and stuff like that, and all the there's nothing shown about this enzyme in this process. What I know is that
because these DNAs will be floating, and is there any chance of this DNA getting within the cells once again, or let's say in the nuclear environment, and then sponging the whole transcription factor because the DNA is intact, right?
Yeah. There are some people like that are saying that these DNAs 1L3 has a nuclear translocation signal and can even go to the nuclei and play a role there. But these studies like are implicating these DNAs 1L3 as basically involved in the fragmentation of DNA during apoptosis. Within the cells, like usually it's an inactive form, and it gets there's a signal peptides that is on its end terminal, and this signal peptide is mostly removed during the secretion process, like after going to the Golgi stuff. So intracellular function, I don't know much about it. I don't know implication of this enzyme in controlling like the transcription and stuff
What I'm suggesting is that the after effect of the DNase 1L3, which would, if we knock it out, then there will be a bigger fragment of the DNA. The resultant bigger fragment could get into the cells and then kind of act as a sponging for the factors that you need for maintaining.
It's an interesting concept. Like I never thought of that, and thank you for the suggestion. But yeah, so this is something that we haven't looked. Like what is known is that bigger fragment, at least, of DNA for B cells are more immunogenic than smaller fragments for the activation of the B cells. This is what we know about like these things. They will be easily recognized by like there's more recognition by BCR receptors. But other than that, like these fragments, how they can enter the cells and modulate as a sponge, like and play a role there. Like, and that's a very interesting concept. Like I have no clue, and it's something that we might be interesting to looking at. Thank you for the suggestion.
Maybe Arindam, Dr. Arindam Talukdar from the other campus.
Yeah, very nice talk. So since you said that both TLR7 and 9 is implicated, so is this DNase 1L3 is known to also cleave RNA, or RNAs are involved along with this, some kind of RNAs?
Yeah, that contributes to our like surprise when we did the double, like we only observed that this effect with the double knockouts. So we don't know. I don't believe that this DNase 1L3 is able. We didn't. I don't know papers that tested this activity of this enzyme with the RNAs. But I think some people. I'm not sure. Like I don't know if it was very characterized. But what was described recently is that TLR7 is like at least TLR9 is specific to how do you call it? RNA, single-stranded RNA. But what is also known is that TLR7 not only recognizes, like what I said before, is not only recognizes like the single-stranded RNA, but it can recognize degradation products of RNA itself, but also DNA, because it can recognize deoxyguanosines and be activated by that. So maybe like that's the case. And then there is also very intriguing data in lupus actually, is that these receptors are very, like, even though we consider that TLR7 and TLR9 do the same in lupus, like a lot of other models of lupus, TLR9 has a protective role while TLR7 has a pathogenic role. And so people are now, like, there are papers now that are suggesting that maybe you need to block like the protective role of TLR9 also in a way, like, so there are papers suggesting that they signal differently, and TLR9 might have multiple signaling partner, at least in B cells, to have some kind of immunoprotective function. But like for us, like, this is still something intriguing, and I don't have a clear answer to your question.
So, and another question is, what about, you said that hydroxychloroquine is kind of giving some kind of effect, but have you thought of any other thing, like hydroxychloroquine, it blocks both 7, 8, 9, so any other therapeutic, any chance?
Like, so so this is the only clinical available drug so far. There is no inhibitors of TLR7 and 9 in the clinics. But those patients are like are on a lot of other things. Hydroxychloroquine is among one of them, like, but there is also rituximab to get rid of their B cells. There is also corticoid immunosuppressive treatments. So far, but like it seems like this at least this hydroxychloroquine works okay in stabilizing a little bit the patients, but it's not perfect as well. It's quite difficult. Like they are like recovering a little bit, but the disease doesn't go away.
Thank you.
Yeah, thank you for your questions.
Yeah. So I had one question.
Yes.
So do these microparticles, do they activate only through MyD88 and type 1 interferon pathway, or do they also activate the pro-inflammatory, the NF-kappa B pathway? So why I'm asking this question is because when you knock out IFNAR and PDCs, so you see like this partial phenotype. So is it possible that the other pro-inflammatory pathway may be activated and pro-inflammatory cytokines are activating the T cells, which may be activating the B cells?
So yeah, we don't know. It's actually very difficult to see the immunogenic capacity of these microparticles in vitro. When you do that, I don't know. I tried some experiments like to find out if DCs get activated by these things, and it's quite difficult. So we haven't looked further because it's difficult to do these experiments and also the amount of microparticles that you should use, what is the physiological relevance of them and stuff like that. So I don't know. So it's something like, I don't know what are the pathways that are induced there a lot. So but what we believe, like, why so there is surely other cytokines that may be involved, and there is a follow-up story that's coming with the impact of IL-6 in our model. So this is so NF-kappa B induced. So, but it's just in some collaboration with something that's happening in the gut microbiota. So this is something that's going to come out soon, and. Why we believe there is an intermediate phenotype of this IFNAR deficiency, a PDC deficiency, because we believe that most of the effect is happening on the B cells directly, and if interferon is and PDCs are maybe important, like in in keeping these B cells, like, autoreactive B cells and plasmacytomes better alive or more stable and stuff like that. But they will be always produced like these antibodies are even enough, like they will be produced because there is a chronic stimulation of your B cells because there is a chronic availability of a ligand. Yes.
So you did not show what kind of difference was there when you are getting rid of PTCs. So was the autoantibodies present in the first few months and then it was not affecting renal pathology or things like that?
I don't
So
have
what-
it here. Okay. So this is Bordeaux. I don't have it here. So what we basically saw is that when you look 5, 10 weeks after the birth of these mice, the amount of anti-DNA antibodies is the same.
Okay.
We did chromatin antibodies is the same. When you continue in age, your other guys are like your knockout mice are increasing, but the haplodeficiency of PDCs is kind of stagnating. Like
Okay.
it's reduced at this point. And
So this is because of IFNAR potentiation of extrafollicular response?
we believe that. We believe that. And so at this time point, so when you sacrifice the mice at the 60 weeks of age, so 1 year or a little bit more than a year, so we didn't see that the splenomegaly was reduced in PDC deficient mice. We don't see because there's usually at the end some T cell activation and B cell activation.
So maybe if you look at the BCR,
yeah.
Will these mice have more somatic hypermutation? Maybe the initial autoreactivity is because of GC.
Yeah,
and potentiation is because of
So we haven't seen,
extrafollicular, right?
maybe we haven't seen that. Yeah, for sure. But then but if you have your TLR7 knockout mice, it's abrogated at least so it's abrogated from the birth. The TLR7, so they will not have germinal center reaction, never. So you would believe that it's this germinal center knockout.
Okay. So you had that control.
But it's kind of hard, to we cannot fully exclude because TLR7 knockout don't have germinal center in the spleen and certain lymph nodes, but in the mesenteric lymph nodes, those germinal centers are present. So so it's we are not pushing the message that it's only extrafollicular like this. We saw extrafollicular, and we see less somatic hypermutation, but and we never see accumulation of germinal centers of these mice. So these are stuff that's going against.
Have you looked into them early enough?
Yeah, maybe not. I haven't looked early.
So there is a chance that early enough.
Yeah, I know why you're suggesting that because our paper was showing that PDC deficiency in other lupus models of mouse were reducing the size of germinal centers, and those mice had a reduced signatures of germinal centers. So that was a surprise for us too. But in this case, was a little bit different. It's a different model.
No question. Okay.
It's okay. I'm sorry.
But we are also lost the time.
Sorry about the time. Yeah. So this is Bordeaux, guys. So whenever you want to come, you are more than welcome. For Beta came, everybody can come and apply to. It's really cool. If these exchange programs are existing and this collaborative grant with the CEFIPRA. So this is a great opportunity, and for me, I'm really happy to be here, and it will and to receive students and friends from India. It's really a privilege for us. So do not hesitate.
Yeah. So do look into CEFIPRA opportunities, and your mentor can definitely forge collaborations. I always tell all my friends to go for CEFIPRA, but also for the students, you can actually have this Raman Sharpak fellowship. Right? So that's a good opportunity to get exposed to other kinds of labs. And thanks a lot,
Thank
Vanja,
you very much
for a very nice and kind of descriptive talk that everyone could understand, I hope. I mean, that was really nice that the field is little esoteric, but still you actually presented it really well so that people get a kind of taste of what you are actually working on. Thanks for being here. Thanks all the audience that have come offline and online. Thank you so much for attending this talk. And thanks a lot to CEFIPRA for making it happen. And so I hope other CEFIPRA research collaborators will also come up with such talks which can be made public, and we get to know what kind of research is being done by this CEFIPRA collaboration. Thank you so much, everyone. Thanks.